Okay. So good evening to one and all. Today is my second session that is based on the photosynthesis topic. So we all know about photosynthesis and what exactly this mechanism and how this mechanism occurs. So we all know that this during the evolution, the previous environment of the earth was in a reducing state. So but so this plant, so the organisms or some another components that they have showed some adaptation and modification. So this first photosynthetic organism, they have probably evolved in the early evolutionary history of life, and shows that the reducing agents such as hydrogen and electron. So the cyanobacteria, which is responsible for the photosynthesis, and they have the biochemical capacity to use water as a source of electron in the photosynthesis, and removed a primary, and they are referred as a primary producer. So we will discuss how this mechanism occur, and what are at which location this mechanism takes place, how many pigments are involved, what are light reactions, where this asymmetry power are going to be used, some concept based on C4 pathway and photorespiration, and some factors which affects the photosynthesis. So this is the location where this photosynthesis takes place. This is a dorsiventral section of the leaf, and this is the enhanced structure of the chloroplast, in which this grana are present, and these are the stacks of coin, which represents the single unit of these stacks of coin are known as thylakoid, and in this thylakoid membrane only the light reactions takes place. This grana, one granum and another granum are connected by a small interconnected bridge known as stromal lamellae. This is the enhanced structure of the chloroplast. We all know that photosynthesis takes place in the chloroplast, and this the primary pigments of chloroplast is a chlorophyll molecule, and such pigments, when they forms a unit, and work together and forms a light harvesting complexes, which helps to capture the light and transfer that energy to the reaction centers. This is the summary of today's session, in which the light reactions takes place in the thylakoid membrane, and our prime objective of light reaction is to produce an asymmetry power, that is ATP and NADPH, which is rich in energy. So this ATP and NADPH that we are going to use in a Calvin cycle, that forms a series of reactions by which this sugar molecule that takes place, sugar molecule formation. And one another phenomena is the light reaction, through which one important mechanism has been associated, that is O2, formation of O2. So these are some of the pigments which are involved in the photosynthesis process. There are two types, principal and the accessory. So this each and every pigment that we are going to discuss in a detail. So these are chlorophyll A, bacterial chlorophyll. The accessory pigments are chlorophyll B, C, D, carotenoid, and phycobilins. So first, this chlorophyll A. We all know that the chemical formula of this chlorophyll A is C55H72O5 and 4 Mg. You can see the molecular structure of this chlorophyll A consists of central magnesium ion, which surrounds the 4 nitrogen atom, and the structure is known as tetrapyrrole or a porphyrin structure. You can see this hydrophilic pyrrole head and hydrophobic phytol tail. These two are embedded into the thylakoid membrane, and this 4 pyrrole molecule, you can see 1, 2, 3, 4 pyrrole molecule. They are linked together by a methane group, CH3, 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 CH3. And this maximum absorption takes place at red and blue region, and this chlorophyll A reflects the green region of spectrum. So that's why most of the plant appear green in color. This is the carotene, and these are some of the pigments and the resultant color. Carotene, which is orange, and xanthophyll, which is yellow pigment, pheophytin A, which is gray brown, pheophytin B, yellow brown, chlorophyll A, which is blue green pigment, chlorophyll B, yellow green pigment. So another principal chlorophyll pigment is a bacterial chlorophyll, which its chemical formula is C55H74Mg N4O6, and this can be found in all purple and green sulfur bacteria. The organism that contain bacterial chlorophyll conducts photosynthesis to sustain their energy requirement, but they do not produce oxygen as a byproduct. So the spectrum which they absorb is the wavelength of the light observed by this bacterial chlorophyll are not fall under like the plant or a cyanobacteria, means they absorb the infrared region of the spectrum. And the another one is phycobilins. Phycobilins, they are also a light capturing agent and found in cyanobacteria. Phycobilins are of two types, phycoerythrin and phycocyanin, which also present in the cyanobacterian red algae. And those two components are also water soluble. And these phycobilins fluoresce at a particular wavelength, and they are used in a research as a chemical tags. And the another one is carotenoid. This carotenoid serves as a two key role in plants and algae. The first one is they absorb the light energy for the use in photosynthesis, and they provide photoprotection. This carotenoid, also called as tetraterpenoids or a yellow, orange, red organic pigments, and they are produced by plants and algae. And the accessory pigments. The another role of these accessory pigments is to protect the chlorophyll A by photooxidation. And this the structure formula of carotene is C40H56, and the maximum absorption takes place at violet region. And this is the structure of xanthophyll, which has OH group at the benzene ring. This is the structure of xanthophyll, which forms lutein. These are the spectral region at which the different pigments absorbs the different region of light. And so this light is absorbed from the entire visible spectrum when the accessory pigments are combined with the chlorophyll A and chlorophyll B. The structural difference between chlorophyll A and chlorophyll B is the presence of aldehyde group at the side chain of second carbon and the methane group in case of chlorophyll A. Only difference occur between chlorophyll A and chlorophyll B is the presence of aldehyde and methane group. These are some of the differences. Different pigment occurs in the leaf, which reflects this blue color shows the presence of phycobilin, and this as the leaf gets deteriorate. As the leaf gets older, this chlorophyll deteriorate, and these chlorophyll pigments are replaced by this carotenoid and xanthophyll. So now this was till here it was all about the pigments. Now we will see how these pigments captures the light. We know that these pigments are present in the PS systems. So we should know the first of all we should know the structure of the thylakoid lumen. It's a typically bilipid layer in which 5 supramolecules are embedded, that is PS1, PS2, light harvesting complex, coupling factor, and cytochrome B6F. So this PS1 and PS2 shows a large amount of pigment, and they capture the light, and those pigments which does not become the member of the PS1 and PS2, they are scattered in the thylakoid membrane and forms a light harvesting complexes. Right. So first we will discuss about the PS1 and PS2 system. So when the light falls on the PS1 system, this accessory pigments capture the light energy in the form of photon and transfer that energy to the reaction center. This reaction center, when they are overloaded with the energy, the electron will get excited from the ground state to the excited state and escapes from the photosystem 1, and this electron primarily accepted by the primary electron and then transferred to the mobile carrier. Those are ferredoxin and plastoquinone, and then to the cytochrome B6 complexes. So the energy rich electron has been harvested for the formation of ATP here, and then this electron again comes in their ground state and then return to their own center. So the voids which has been formed by the excitation of the electrons are again filled by those electrons only which are now in a ground state. Now another system is PS2 system. The same mechanism occurs in this also, but the only difference is that when the electron excites and the primary electron accepts that electron, the same mechanism happens, that the formation of ATP. But the electron which is now in a ground state, it won't come back into its own reaction center, because during the capturing of light, the simultaneous process also occurs, that the photolysis of water in PS2 system. So if we take two molecule of water, means 2H2O, it will form 4H plus and 4 electron plus 2O, which will rearrange and forms oxygen molecule. So this 4H plus and 4 electron will occupy the voids in the reaction center, which has been produced by the excitation of these electrons in the presence of Mg plus ions, right? So this is the exact mechanism which occurs during this PS2 system. It's a Z scheme. It looks like N, but it's a Z scheme, and it's a unidirectional flow. And this process is this photophosphorylation is known as non-cyclic photophosphorylation, because this cycle has not been completed yet. So we have discussed till here. After this, when the cytochrome B6 complex harvests the energy for the production of ATP, these electrons, the energy deficient electron again occupy their space in the PS1, because they won't come their own reaction center, because their space has been occupied by this 4 electron. So there is no further space for these electrons. So they will occupy the space in PS1 system, and again this PS1 system, when it excites, the mobile carrier bounds this electron and forms and transfer this electron to the NADP, which is present in the stromal portion of the chloroplast, and gets. So we know that this electron has been used. Now what about this 4H plus? So this 4H plus will bind with NADP and forms NADPH. The molecule will remain. If we take 3 molecule of NADP, it will form 3 molecule of NADPH, or 4 molecule, then 4 molecule of NADPH. So this process is independent, and this process only takes place in PS1 system, which takes place at 700 nanometer wavelength. So this is the basic difference between cyclic photophosphorylation and non-cyclic photophosphorylation. This involves PS1 only, and in non-cyclic both PS1 and PS2 takes place. The movement of the electron is cyclic, whereas in non-cyclic photophosphorylation this movement is unidirectional. Only ATP is formed. Both ATP and NADPH are formed. Oxygen is not evolved. Oxygen is evolved. This process takes place in photosynthetic bacteria, whereas this process takes place in higher plants, green plants. Photolysis of water does not occur, and photolysis of waters occurs. Now this is the process through which we can understand how this ATP has been formed. So this is the chemiosmosis theory, in which the movement of ion across the semi-permeable membrane bounds the structure down their electrochemical gradient. For example of this, it would be the formation of adenosine triphosphate by the movement of hydrogen H plus ion across the membrane during the cellular photosynthesis. So we know that this is the structure of the thylakoid membrane, and in which it contains 5 supramolecules, right? So our prime objective is to create a potential gradient between the stroma and the lumen. So how we can produce this gradient? So first of all, we know that this PS2 system has the activity of photolysis, right? So it after the breakdown of H2O, it forms. We can see here after the breakdown of H2O, it forms 4H plus 4 electron plus 2O, which will rearrange itself and form O2 molecule, right? So one reason is this, that the proton gradient increases by the photolysis of water. Another reason is by the PQ, plastoquinone. What happened when this plastoquinone accepts the electron from the PS2 system? It also accepts the hydrogen atom from the stroma and forms PHQ. So when this plastoquinone come across this membrane, so it releases H2 molecule into the lumen and again convert it into PQ. So this H2 has been released in the lumen, which also responsible for the increase in the proton gradient from stroma towards the lumen, right? And the third reason is this NADP. NADP accepts the H plus ion which is present in the stromal portion of the chloroplast and get reduced into NADPH, right? So this process takes place only in PS1 because this PS1 system has the NADP reductase enzyme, right? So this was all about the proton gradient through which we can produce our ATP. So now we are clear that this proton gradient has been increased due to three reasons. Now what will happen? Our ultimate objective is to produce ATP. So this ATP, so this H plus ion move from coupling factor which has ATP synthase enzyme, and in the presence of ATP synthase enzyme, this ADP plus inorganic phosphate and 1H plus forms ATP molecule, right? So as the concentration increases, this H plus ions diffuses from higher concentration to lower concentration, and as a result we will get our desired object, that is ATP, which is energy rich component, right? So still here it was all about the preparatory phase of the photosynthesis. We have produced our assimilatory power that we are going to use in for the formation of our food, that is glucose. So now we will discuss about Calvin cycle. We know that this Calvin cycle is a part of photosynthesis which occurs in two stages. In the first stage, these chemical reactions use energy from light to produce ATP and NADPH, and in second stage, this Calvin cycle or a dark reaction, carbon dioxide and the water molecules are converted into organic molecules, that is glucose, right? So this Calvin cycle, also known as dark reaction, and this reactions doesn't actually occurs in the dark, but or during night time, but this reaction is require the reduced NADP which comes from the light dependent reaction. So there is no direct involvement of light. That's why they call this reaction as a dark reaction, right? So our first objective is our first process in Calvin cycle is to fix the carbon. First phase is carbon fixation, second is reduction, and third is regeneration of the ribulose, right? So first step is carbon fixation. So how we will fix the carbon? So the carbon dioxide. So you first of all we should know that RUBP, that is ribulose by 5-phosphate, right, which is 5 carbon compound. You can see this black balls which represents the carbon, 5 carbon compound with at which 5 carbon 1 phosphate group has been attached. So this ribulose, we know that ribose is a pentose sugar and the isomer of ribose is ribulose. So it's nothing but the ribulose is attached with phosphate group. That's why we call it as a ribulose 5-phosphate, right? So this ribulose 5-phosphate act as a substrate, and this enzyme Rubisco has a dual nature, that is carboxylation and oxygenation. So this Rubisco, when the concentration of CO2 is increased in the plant, this Rubisco has a greater affinity towards the CO2 and forms and proceed with the carboxylation. So this Rubisco, in the presence of CO2, fixes this 5-carbon compound and converts this 5-carbon compound into into 3 carbon of 2 molecule, that is 3 PGA. You can see this 1, 2, 3, 4, 5 carbon compound, which is attached with one, which is now fixed with CO2, that is 1 carbon compound, 1 carbon, which has 1 carbon. So after the fixation, the molecule which has been produced, which contain 3 carbon compound, and that is 2 molecule, means 2 molecule वाला 3 carbon वाला 2 molecule बनेगा, that is phosphoglycerate. So this or we can say 3 PGA. 3 is the position at which this phosphate group has been attached. You can see 1 2 3 and 3 carbon पे एक phosphate group है और इसका 2 molecule बना है, एक ये और एक ये, राइट? और यहाँ पे 5 कार्बन थे जिसके साथ कार्बन डाइऑक्साइड को मिक्स किया गया, तो 6 कार्बन टोटल हुए. तो 6 कार्बन हुए, राइट? सो जिसके 2 मॉलिक्यूल बन रहे हैं, दैट विल लीड्स टू 3 कार्बन कंपाउंड. 2 into 6 कार्बन और 2 मॉलिक्यूल बना. During that time only, what happened? We know that during एसिड का जब रिडक्शन होता है तो इट फॉर्म्स एल्डिहाइड, राइट? सो सेकेंड फेज इज रिडक्शन. टिल हियर वी हैव फिक्स्ड अवर कार्बन, राइट? और उसको 2 मॉलिक्यूल में कन्वर्ट कर दिया, दैट इज 3 PGA. अब 3 PGA का फर्दर रिडक्शन हमें करना है, राइट? सो दिस 3 PGA का रिडक्शन कैसे होगा? एक ये जो 1,3 बिसफॉस्फेट है, इसमें ATP, व्हेन वी ऐड दिस ATP एसिमिलेटरी पावर, जो कि हमने लाइट रिएक्शन में बनाई थी, दिस ATP इन्वॉल्व एंड रिलीजेस ADP. और क्या हुआ? एक फॉस्फेट ग्रुप इसको दे दिया. सो ये क्या हो गया? 1,3 बिसफॉस्फोग्लिसरेट. 1 नंबर के कार्बन पे एक फॉस्फेट है और 3 नंबर के कार्बन पे एक फॉस्फेट है, दैट फॉर्म्स 1,3 बिसफॉस्फोग्लिसरेट. 2 फॉस्फेट एंड ग्लिसरेट, राइट? अब इसका हमें रिडक्शन करना है. वी हैव ऑलरेडी प्रोड्यूस्ड द रिड्यूसिंग एजेंट ड्यूरिंग लाइट रिएक्शन, दैट इज NADPH, राइट? सो दिस NADPH, ये क्या होगा? इसका ये रिड्यूसिंग एजेंट है, ये इसको रिड्यूस कर देगा. इसने रिड्यूस कैसे किया? इसमें ग्लिसरॉल्डिहाइड 3 फॉस्फेट है. अब इसमें क्या होगा? ये यहाँ पे जो है, वी शुड ऑलवेज कंक्लूड दैट यहाँ पे हम 2 मॉलिक्यूल लेके आपको चलना है. 2 मॉलिक्यूल इसका, 2 मॉलिक्यूल इसका, राइट? अब इसका भी 2 मॉलिक्यूल है, मीन्स इसके पास 6 कार्बन हुए. 2 मॉलिक्यूल है, एक मॉलिक्यूल में अगर 3 कार्बन है तो 2 मॉलिक्यूल में 6 कार्बन है, राइट? सो यहाँ पे भी जो बनेगा वो 3 कार्बन का 2 मॉलिक्यूल बनेगा, मीन्स जो ग्लिसरॉल्डिहाइड 3 फॉस्फेट है, इसके पास भी 6 कार्बन हो गए टोटल. सो ड्यूरिंग दैट प्रोसेस, रिडक्शन प्रोसेस, जो कुछ मॉलिक्यूल है ग्लिसरॉल्डिहाइड 3 फॉस्फेट के, वो रीजनरेशन स्टेप में वापस चले जाएंगे. बिकॉज अगर हमको, इफ वी वांट टू कैरी दिस साइकिल कंटिन्यूअसली, तो यहाँ पे से RUBP का भी एक्सटिंक्शन नहीं होना चाहिए और CO2 का भी नहीं होना चाहिए, राइट? तभी ये प्रोसेस चलती रहेगी. सो इसलिए हमको रीजनरेशन स्टेप भी जरूरी है. सो 1 कार्बन विल फॉर्म द 1/6 part of the glucose, and the 5 carbon compound will come into the regeneration step and forms the ribulose 5-phosphate. ये जो 1 कार्बन जो यहाँ पे से जो निकलेगा, तो we know that the fixation of 6 molecule of CO2 and 6 turn of Calvin cycle are required for removal of 1 molecule of glucose from the pathway. Means यहाँ पे जब हम सिर्फ एक मॉलिक्यूल ऑफ CO2 ले रहे हैं तो उसमें हमको क्या हो सकता है कि 1/6 पार्ट ऑफ ग्लूकोज जो होगा वो बनेगा, राइट. सो दिस CO2 जब हम एक और वन कंसेप्ट इसका ये भी है कि जब एक मॉलिक्यूल CO2 पे जब 6 टर्न पे ग्लूकोज एक मॉलिक्यूल बनेगा तो 6 टर्न हमें चाहिए, तभी हमें C6 मिलेगा, राइट. एक मॉलिक्यूल, समझ रहे हो? हेलो हेलो
हेलो. यस यस, बोलो भाई, बोलो सब.
हाँ हाँ बोलिए बोलिए.
6 वी नो दैट कि जो एक कार्बन है उससे वो जब पूरे एक साइकिल कंप्लीट हो रहा है तो वी कैन कंसिडर कि 1/6 पार्ट जो है वो ग्लूकोज का बनेगा, क्योंकि यहाँ पे हमें 6 टर्न चाहिए कैल्विन साइकिल के एक मॉलिक्यूल ग्लूकोज बनाने के लिए, राइट. सो जब 6 कार्बन होगा तो अल्टीमेटली हमें एटीपी कितना जाएगा और एनएडीपी कितना जाएगा, तो इन कितना हुआ जब हम 6 मॉलिक्यूल का इन एंट्री करेंगे तो एक मॉलिक्यूल ग्लूकोज मिल रहा है हमें, और 18 एटीपी इसमें यूटिलाइज हो रहा है और 18 एडीपी निकल रहा है, वो हम अभी डिस्कस करते हैं. और 12 NADPH अंदर जा रहा है और 12 NADP बाहर आ रहा है. अब ये कैसे हो रहा है आप लोग यहाँ पे देख सकते हो, यू कैन सी एटीपी. यहाँ पे आपको कंसिडर करना है कि एक कार्बन पे जब 2 एटीपी जा रहा है, क्योंकि यहाँ पे 2 मॉलिक्यूल हमने बनाया है तो हर मॉलिक्यूल पे अगर एक एक एटीपी दे तो यहाँ पे भी हम 2 मॉलिक्यूल ही लेंगे एटीपी का. सो दिस 2 मॉलिक्यूल ऑफ एटीपी यहाँ पे जा रहा है, तो 2 into 6 जितने भी मॉलिक्यूल है उसको सबको 6 से इंटू कर देना है जब हमें ग्लूकोज एक मॉलिक्यूल बनाना होगा तो. सो 6 कार्बन पे 1 ग्लूकोज मिल रहा है, तो 6 into 2 जब एक कार्बन पे 2 एटीपी जा रही है तो 6 कार्बन पे कितनी जाएगी, 12, राइट. 12 एटीपी यहाँ पे हो गई और 12 NADP यहाँ से बाहर ADP बाहर निकला, राइट. अब यहाँ पे एक मॉलिक्यूल पे एक NADPH, एक मॉलिक्यूल को एक NADPH रिड्यूस कर रहा है तो 2 मॉलिक्यूल को कितने NADP रिड्यूस करेंगे, तो 2 मॉलिक्यूल ऑफ NADPH जो है वो रिड्यूस करेंगे 2 मॉलिक्यूल ग्लिसरॉल्डिहाइड 3-फॉस्फेट को. तो यहाँ पे भी कितने मॉलिक्यूल हमें NADPH के चाहिए, तो 12 मॉलिक्यूल हो जाएंगे, बिकॉज 6 into 2 12. अब बचा ये कि आपको यहाँ पे यू हैव टू फोकस ऑन दिस रीजन, इन दिस रीजन, राइट. वी नो दैट कि यहाँ पे ग्लिसरॉल्डिहाइड 3-फॉस्फेट का 2 मॉलिक्यूल है, मीन्स इट कंटेन 6 कार्बन, राइट. सो 5 कार्बन तो रिबुलोज 5-फॉस्फेट बनाने में चले गए और एक कार्बन यहाँ आ गया, तो ये जो 5 पर यहाँ पे 5 कार्बन तो जो है उसके साथ रिबुलोज 1,5 बिस फॉस्फेट है, राइट. सो एक एक्स्ट्रा फॉस्फेट कहाँ से आएगा, सो यहाँ पे भी एटीपी का यूटिलाइजेशन होगा जिसमें से एटीपी अंदर जाएगी और एडीपी बाहर आएगा, उसमें से एक इनऑर्गेनिक फॉस्फेट ग्रुप जो है वो यहाँ पे आ जाएगा. यहाँ पे आप देख रहे हैं एटीपी अंदर इसका इन्वॉल्वमेंट हुआ है जिसने एक अपना फॉस्फेट ग्रुप रिबुलोज 1,5 बिस फॉस्फेट रिबुलोज 5-फॉस्फेट को दे दिया, तो वहां पे जो फर्स्ट कार्बन पे है एक और एक फॉस्फेट ग्रुप आ गया, तो ये क्या बन गया, रिबुलोज 1,5 बिस फॉस्फेट, राइट. इतना समझ में आया? नाउ वी विल डिस्कस, दिस वाज ऑल अबाउट दिस C3 साइकिल. सो अल्टीमेटली यहाँ पे 2 एटीपी और यहाँ पे 1 एटीपी, तो कितना हुआ टोटल 3 एटीपी का इन्वॉल्वमेंट हुआ, और इसमें से निकले कितने, 3 एटीपी का इन्वॉल्वमेंट हुआ तो 6 टर्न जब होंगे तो कितना होगा, 6 कार्बन डाइऑक्साइड जब होंगे तो 6 into 3 18 एटीपी हो गया, और 18 ADP बाहर आया। ये लास्ट वाला जो ADP है जो बाहर जा रहा है, ओके। और 12 NADPH जो हमने यहाँ पे ऑलरेडी डिस्कस कर लिया। नाउ वी विल डिस्कस अबाउट C4 साइकिल। नाउ C4 साइकिल में क्या होता है? C4 साइकिल does not show photorespiration. That we will discuss later कि what exactly this photorespiration is. But C4 साइकिल के पास जो डेफिशिएंसी C3 साइकिल की है ना, उस चीज को C4 साइकिल ओवरकम कर रहा है। अब कैसे कर रहा है? How this C4 cycle overcome the deficiency or the disadvantage of C3 cycle? Because so first we will discuss what is C4 cycle, then we will discuss how. Okay. So when this CO2 comes inside from the atmosphere into the leaf, so जो C3 cycle में जो first carbon fixation का जो enzyme था, that is ribisco. But here that is PEP carboxylase. So this PEP carboxylase is a 3 carbon containing compound which mix with the CO2 and forms 4 carbon compound, that is oxaloacetate or oxaloacetic acid, कुछ भी बोल सकते हैं. So this oxaloacetic acid is a 4 carbon compound which will reduce into malic acid or a malate, जो कि also 4 carbon compound ही है. So यहाँ पे जो reduction होगा oxaloacetate का into malate, it forms due to the NADPH. यहाँ पे NADPH का involvement होगा, तभी ये OAA reduce होगा malate में और malic acid में. So when this malic acid gets diffused from this mesophyll cell to the bundle sheath cell, so again this malates get break down into 3 carbon compound, that is pyruvate and 1 CO2. अब यहाँ पे major objective of this C4 cycle is to transfer the CO2 safely towards the C3 cycle, where not towards the C3 cycle, towards the region where this CO2 has been safely utilized by the C3 cycle. क्योंकि अगर ये C3 cycle मेसोफिल सेल के क्लोरोप्लास्ट रीजन में हो रहा होता, सो वहां पे स्टोमेटा जो है विच इज नियर बाय द मेसोफिल सेल, सो वहां पे गैशियस एक्सचेंज और वाटर का एक्सचेंज भी होता रहता और ऑक्सीजन और CO2 भी होता रहता. वी नो दैट दी जो CO2 का जो कंसंट्रेशन है इन द एटमोस्फियर इज लेसर देन द O2, right? So वहां पे ऑक्सीजन भी होता. So the rubisco, as the concentration of the O2 increases, rubisco has the affinity to bind with the O2 also, because it also contains the oxygenation capacity also. So जो rubisco का जो main function है वो CO2 या O2 के concentration पे डिपेंड करती है. इस चीज को नोट कर लो कि O2 का कंसंट्रेशन अगर ज्यादा होगा तो रुबिस्को जो है वो ऑक्सीजनेशन करेगा and that leads to photorespiration. That is a doubly wasteful process that we will discuss in a later slides. First here we are discussing the C4 cycle. इसमें PEP carboxylase क्यों आया? Because इसका affinity CO2 के ही towards है. So safely CO2 मेसोफिल सेल से बंडल शीथ सेल में आ जाए, इसका प्राइम ऑब्जेक्टिव है. और C4 साइकिल और C3 साइकिल में स्लाइटली डिफरेंस इज द लोकेशन ओनली. और इसके अंदर C3 साइकिल तो होगा ही, बट दिस साइकिल, दिस रिएक्शन ऑक्सैलोएसीटेट और मैलेट का कन्वर्जन, this is the additional point in this C4 cycle. This particularly large cells around the vascular bundle we have already discussed in the anatomical structure of the leaf. So we know that the exact location where this bundle sheath cells are present, and this bundle sheath cells are very far from this stomata. So this particularly large cells around the vascular bundles of the C4 plant known as bundle sheath cells. You can see this bundle sheath cells, right? And the leaf which shows this kind of anatomy are known as Krenz anatomy, right? So this Krenz means the breadth, and it's a reflection of the arrangement of, it's just the arrangement of the cells which shows this anatomical structure, right? सो आफ्टर दिस जो ये जब पायरुवेट हो गया वो दूसरे साइकिल में वापस चला जाएगा एंड दिस CO2 अगेन कैरीज दिस कैल्विन साइकिल प्रोसेस, राइट. सो ये प्रोसेस होगा बंडल शीट सेल में. नाउ वी विल सी द differences between C3 and C4 cycles. So different between C3 and C4 cycles are the first stable compound in C3 cycle is the 3 carbon containing compound, that is 3-PGA or 3-phosphoglyceric acid, right? Whereas in C4 it's a 4 carbon containing compound, that is oxaloacetic acid. Now first observation is in C3 cycle. The C3 cycle was first observed by Calvin, or C4 cycle is has been observed by Hatch and Slack. Alternative name of these cycles is Calvin cycle and Hatch-Slack cycle. And the presence, the C3 cycle is found in all plants, whereas the C4 cycle is found only in the C4 plant like sorghum and maize. And the primary carbon dioxide acceptor in C3 cycle is rubisco. This is the substrate. And the C4 cycle, the primary carbon dioxide acceptor is PEP. Carboxylase enzyme, in C3 cycle, this carboxylase enzyme is rubisco in C3 cycle, and carboxylase enzyme are PEP, carboxylase and rubisco in C4 cycle. ऐसा नहीं है कि C4 cycle में जो रुबिस्को वाला जो नेचर है वो नहीं होगा, बिकॉज वी हैव सीन दैट यहाँ पे कैल्विन साइकिल जो थॉरली जो कैल्विन साइकिल है वही ही रन होगा, बट इसका जो पिकअप पॉइंट है कार्बन डाइऑक्साइड का वो रुबिस्को नहीं है, पेप कार्बोक्सिलेज है इन केस ऑफ C4 साइकिल. सो नाउ द कार्बन फिक्सेशन. कार्बन फिक्सेशन में अ सिंगल कार्बन फिक्सेशन अकर्स इन C3, whereas C4 cycle में double carbon fixation can take place. Efficiency in carbon fixation is lesser in C3 cycle as well as slow, whereas the carbon fixation is more efficient and fast in C4 cycle. Requirement of the carbon fixation. The C3 cycle, this fixation of the single carbon molecule requires 3 ATP and 2 NADH, and the fixation of single carbon fixation requires 5 ATP and 3 NADH in case of C4. Types of chloroplast. C3 cycle में granular chloroplast होता है involved in the C3 cycle, and C4 cycle granular and agranular chloroplast involved in the C4 cycle. Now Kranz anatomy in the leaf. So this C3 cycle is Kranz anatomy is absent in the leaves of C3 cycle and present in the C4 cycle. C3 cycle is carried out by the mesophyll cells alone, and this C4 cycle is carried out by both mesophyll cells as well as the bundle sheath cells. Optimum temperature of C3 cycle is 20 to 25 degrees Celsius, whereas optimum temperature of C4 cycle is 30 to 45 degrees Celsius. 1 second sir, ये बैटरी लो हो रहा है, 1 सेकेंड. Now again, at lower carbon dioxide concentration, this C3 cycle is unable to proceed at low carbon dioxide concentration, whereas C4 cycle can proceed at very low carbon dioxide concentration. And effect of oxygen. When we talk about the effect of oxygen, C3 cycle is inhibited by oxygen by the presence of oxygen. We already know that photorespiration will take place, but in C4 cycle, no inhibition of C4 cycle takes place when the oxygen is present in that plant. So now the sunlight. The C3 cycle can be saturated with sunlight, and C4 cycle does not saturate it with the sunlight. Photorespiration, considerable amount of photorespiration is observed in C3 cycle, whereas negligible amount of photorespiration is observed in C4 cycle. So the conclusive part of this C3 and C4 cycle are the two type of dark reaction that occurs during the photosynthesis, and this C3 cycle occurs in all plants at 20 to 25 degrees Celsius, whereas this C4 cycle occurs only in the C4 plants at 30 to 45 degrees Celsius. And during this C3 cycle, single carbon fixation event has been observed while this C4 cycle, 2 carbon fixation events are observed. Photorespiration occurs during the C3 cycle, but negligible amount of photorespiration takes place in C4 cycle. And the efficiency of C3 cycle is low when compared to the efficiency of C4 cycle, because because this C4 cycle shows the advanced mechanism um, to neglify those kind of things which hinders the photosynthetic yield. And the main difference between the C3 and C4 cycle is the number of carbon in the first stable compound produced in each cycle, right? In case of C3, we already know that the first stable compound is 3 carbon compound, and in case of C4, that is 4 carbon compound oxaloacetic acid. Now, another topic is photorespiration. So we know that photorespiration is doubly waste process. How? That we will discuss. So this photorespiration, also known as the oxidative photosynthetic carbon cycle or C2 photosynthesis, that refers to a process in the plant metabolism where the enzyme rubisco oxygenate RUBP and wasting some of the energy produced by the photosynthesis. Right? Abhi kaise hoga, that we will discuss. So we know that this rubisco shows a dual nature, carboxylation ka bhi and oxygenation ka bhi, right? So when the concentration of the oxygen increases, this rubisco shows the affinity towards O2 and fixes the RUBP in the presence of O2, and the resultant product that has been produced, that is phosphoglycolate, right? This phosphoglycolate and small amount of 3-PGA plus 2H plus. So इस प्रोसेस को करने के लिए एनर्जी देनी पड़ती है, जो कि that we have already produced in light reaction, right? So इसमें जो कन्वर्जन होगा, इसमें ATP का इनपुट होगा, NADPH का इनपुट होगा, जो कि वेस्टफुल एनर्जी, वेस्ट हमारे जो एनर्जी जो प्रोड्यूस की हुई है उसका वेस्ट कर देगी, एंड अगेन ये जो रिएक्शन है वो यूजलेस हो जाएगा. बट during that time, if rubisco binds with CO2, then we already know that it produces sugar, right? So this is a useful process. Photosynthesis is a useful process and photorespiration is a wasteful process. Now, how this process occurs, that we will discuss. So, firstly, we will start with this ribulose biphosphate. We know that when this RUBP binds with CO2, it forms phosphoglycerate and carry out the process of Calvin cycle and forms by series of mechanism, it forms hexose sugar that is a useful product, right? It's a food for the plant. But what if this RUBP binds with O2? It forms phosphoglycolate, right? So this phosphoglycolate, in the presence of ATP, in the presence of NADH, get reduced to glycolate, and this glycolate again converts into glycine. Glycine में कैसे convert होगा? ये जो है यहाँ पे से जो surrounding medium है उसमें से एक amino group ले लेता है. And we know that glycine is the simplest amino acid. So this glycine has been produced by addition of amino group, and this glycine gets diffuses from this cytosol to the peroxisome, and this glycine again by uptaking the amino group, it again forms this serine. Serine का जो conversion है वो again amino acid जो amino group जो mitochondrial के cytosol में present है उसमें से ले लेगा. And what happened, इसका serine again diffuses from mitochondria to again peroxisome, and it liberates. इसका क्या होगा, अब serine का breakdown हो जाता है. जब breakdown होता है तो it release CO2. CO2 का release जो होता है, that will again use in the Calvin cycle. But what happened, this serine again converted into hydroxypyruvate by series of some enzymes, and this hydroxypyruvate again converted into glycerate, that is a 3 carbon containing compound. So this glycerate again diffuses from this cytosol to the कौन सा था साइटोसॉल से यहाँ पे आ जाता है क्लोरोप्लास्ट में and form phosphoglycerate by the addition of ATP, which is a 3 carbon compound which we have again regained by this diffusion mechanism in this photorespiration. So this is a very lengthy process, but what happened, this phosphoglycolate is 2 carbon containing compound, जिसका जब रिडक्शन होगा तो वो जिसका एटीपी के प्रेजेंस में जब उसका कन्वर्जन होता है तो वो ग्लाइकोलेट में बन जाता है, जो कि 4 कार्बन कंपाउंड है. सो अगेन सीरीज ऑफ रिएक्शन होके एक कार्बन यहाँ से निकल गया, एंड ये जो बचा ग्लिसरेट जो कि 3 कार्बन कंपाउंड है, that will again diffuses from cytosol to the chloroplast and again participate into the Calvin cycle and forms hexose sugar, which is a useful process. But some researchers have found that this photorespiration is beneficial for the conversion of nitrate to the nitrite. So nature has favored this mechanism because of some reason only. Right? So now this was all about how this mechanism and the processes takes place. Now, how this photosynthesis means, what are those factors which affects the rate of photosynthesis? So first we will discuss about the light. It is one of the major factors that affect the photosynthesis, and the photosynthesis begins at the low intensity of the light and increases till it at the maximum at the brightest time of the day. At the highest intensity, the temperature of the plant increases, which leads to the increased transpiration in the plant, that leads to the closing of the stomata, means ultimately the reduction of the exchange of gases, right? And the another one is duration. Duration means the longer the plant is exposed to the light, the longer the process of photosynthesis will continue and takes place, right? As the temperature increases, the plant remains balanced and photosynthesis will occur. But the carbon dioxide, we know that the atmospheric carbon dioxide concentration is 0.03%, and among other gases, and the plant takes up the carbon dioxide from the air. But since the amount of CO2 in the air is very less, it acts as a limiting factor for the photosynthesis. Now the another one is temperature. As the rate of photosynthesis increases with increase in the temperature, now we know that each and every plant have their optimum level at which they shows their best production efficiency. So beyond the optimum level on both the sides of normal range, the enzymes are deactivated, right? Enzyme require the optimum temperature for their better efficiency and better work. And so these enzymes are deactivated or destroyed, and photosynthesis will stop if the temperature increases or decreases. So now the water, when the water is considered one of the most important factor affecting the photosynthesis. So when there is a reduced water intake or availability, the stomata begins to close or avoid the loss of any water during the transpiration, and with the stomata closing down the CO2 intake, it also stops, which affects the photosynthesis. Now, the another one is oxygen. The optimum level of oxygen should be maintained. So, if to favor the photorespiration in C3 plant, and the byproduct of photorespiration is CO2, which is essential for the Calvin cycle, right? So somehow this is helpful, but we can't say this is helpful because it utilizes our energy that we have produced in a light reaction. So ultimately, they show some kind of modification. We can consider this photorespiration is one kind of modification. Now, another one is Blackman's principle of limiting factor. That is, this principle states when the process is governed by more than one factor. So the rate of process is governed by that factor which is closest to its minimum value. Means it, for example, if the leaf is exposed to the certain amount of light intensity with a constant temperature, but CO2 is not available in an efficient amount, so the rate of photosynthesis will decrease, right? And so it won't get hindered by, or it won't favor the light intensity or anything else, because CO2 is must. So therefore in case of CO2, in this case this CO2 is the limiting factor. So thank you.
Very informative, sir.
Thank you.